I'm going to start a little off the marine theme here and talk to you about flowers, just because I think phenology and sort of time when flowers first emerge or time when we first see the trees budding or leaves changing, I think that's a sort of seasonal process that's a little bit easier for everyone to get a mental image on. So we think about things like this. You see the first occurrence of flowers, you see a peak in flowering, and then you see the last of the flowers at the end of the season. So one process that's been observed, again, because things like flowers are things that gentlemen scientists have been observing for hundreds of years, we have really long-term records here, and people have noticed changes in this process in response to changing temperatures over time. But what we've, as a community, started to think about more recently is that these flowers are interacting with other components of the ecosystem. So for example, bees pollinating the flowers are going to be changing their behavior and the availability of different flowers to pollinate as the flowers start to change when they're there and when they're flowering over the season. Okay, but this can get more and more complicated as we go on. So the bees are often choosing between similar flowers to pollinate. For example, are these pink flowers going to be competing now for bees between these purple or red flowers, etc. All right, so we've got this growing web of potentially complicating species interactions as we continue to think about potential chain reactions just for this one process that we were so keen to observe easily of flowers changing their seasonal distribution over time. All right, and now another layer of complications. We can think that there are actually a lot of ways that flowers could be changing their response to climate over the season. They could be uniformly just flowering sooner as a response to temperatures, warmer temperatures coming sooner in the season. They could be shifting later, or we could see changes in just the abundance over time without an overall pattern change. Okay, and again, this is going to scale up to affect these different players that I've now outlined in our web of interactions. Okay, and so when we think about these plant pollinator interactions, as they're called, and we other groups have looked at changes in specific interactions between plants and their pollinators, they sort of tabulated why we know this is occurring or not. So we've seen interactions that are lost, so species are no longer interacting with each other because they're no longer occurring at the same time. So those are the orange and gray squares here. Because they're no longer occurring in space, maybe some of the ranges of these species have shifted, and those are the green squares. But what I'd like to highlight is actually, it's a little bit hard to see here, but most of the squares that are colored in are red, and that means that the interactions have been lost and nobody knows why. So we haven't been able to account for that for either of those really obvious mechanisms that we've been studying so far. Species interactions are actually one of the main things that are going to respond to climate change, but it's actually one of the processes that we know the least about, especially in this system, plants and their pollinators that people have studied really extensively for hundreds of years. So just think about all the other systems that have been studied less. Okay. So that's what I'm trying to do, trying to look at differences in each species and their physiology, how they might respond differently to climate stressors, and then how does that affect the ecological function of a community as you add all those things back up. Okay, so we're sort of interested in the ecology, but looking at the physiology as a sort of a lens to view the ecological world from that perspective. So I'm going to talk about a different climate stressor to what Danny just introduced for us, and that's ocean acidification. So we're burning fossil fuels. Most of that, or I should say a large proportion of that, is carbon dioxide or CO2. And that actually dissolves in seawater, which at first we thought was this great solution to our climate problem. The oceans are getting rid of our CO2 for us, and then we realized that that was actually going to be a problem for the ocean as well, because it affects seawater acidity. For those of you who haven't taken chemistry since high school, you don't have to close your eyes. This is just one slide, I promise. But you have carbon dioxide combines with water, and it forms carbonic acid. That's actually the acid that buffers the blood in our own bodies. Okay, so it's a very common acid everywhere. It can lose one proton to make this carbon nutrient called bicarbonate, and it can lose a second to make carbonate. And as pH goes down, as we get more CO2 in seawater, you get less carbonate, more bicarbonate, and then at really extreme levels, mostly just dissolved CO2. Okay. So we care about this because carbon is a resource. Aquatic plants use dissolved CO2 and bicarbonate for photosynthesis. But on the other hand, shelled animals like mussels and corals use carbonate for their shells and skeletons. So I'm going to take you a little bit away from Florida, or very far away from Florida, to the northern Pacific, to a little island off the coast of Washington State. And this is where I've been working for a long time because there are really, for ecology, long-term records here. And I know the people talking about climate history, if you're here, I apologize for referring to this as long-term since the year 2000. But we've got pH of seawater going down pretty steadily since then. And over the range that we see, and I'll show you, there's a lot of variation. This is over spring and summer. So thinking back to the last presentation, there's a lot of variability in these factors even over the day and over the season. But we're seeing a shift here from a system that's dominated by carbonate, good for shells, to a system now that's dominated by bicarbonate, which is less useful for shells, but good for plants and photosynthesis. And this is happening sort of isolated from other changes in the ocean in this particular location. So I've been using it as sort of a natural laboratory. Okay. And so I have some shells from the archival record. This site is on the Makah tribal nation, and so they have archaeological records that they've shared with us, which has been really fantastic. So we have shells from middens that date back to 663 AD, but we'll just refer to them as about 1,000 years AD. Archival shells from researchers who worked on this island from the 1960s to 1990, and then modern shells that I collected that started growing in 1999 through 2015. And we're able to do this because this island, as you can see here, was used as a summer fishing camp by the Makah tribe for hundreds and thousands of years. Okay. So mussels can archive seawater chemistry. They lay down annual growth rings, just like trees. See them a little bit here laying down horizontally. So the first thing that we did was look at the carbon isotope values of these shells. So this, for the purposes that I use it, is just sort of telling you what's going on with inorganic carbon in this region. I wasn't using this to calculate anything like quantitative, but more just to say, have we seen changes over time? So these black dots here, going back to 660 AD, are pretty constant in their level, but we see in the archival shells, starting from the 1960s, we see a change, and we see an even bigger change when we look at the modern shells from the last 15 years. And this is just a zoom in on the modern shells, and so you can see that this is matching that decline that we saw in the pH measurements, okay? So there's something going on that's changing the inorganic carbon cycle here, and I think it's ocean acidification, probably with a mixture of other processes. So we also looked at the animal's response. So we're sort of multitasking with these mussel shells, using them to tell us something about the environment, but also looking at how the animal is responding to that environment. So we looked at thickness of the shell, but that's the thickest part of the shell, so it gives us more resolution to look at. So what we're looking at here are really zoomed in images on that red box. And you can see these long, sort of thin crystals, either blue or green. So calcium carbonate sort of stacks, if you think about it as a bunch of bristles in a paintbrush. And what the colors are showing you is how the crystals rotated in a plane, sort of like this. And so we can see this is pretty uniform. They're all long and skinny, and they're all blue-green, which means that they're all lined up like this, but potentially rotating a little bit like that, okay? So these are two very healthy shells that were collected in the 1970s. So now you can see this has been pretty consistent. These are the midden shells. The calcium carbonate quality is a little less nice, which you would expect because they're over 1,000 years old. The quality, or sorry, these shells again look really similar. These are the same ones I just showed you, and these are shells collected in 2009, and we're looking at growth probably from the mid-2000s. Okay, and I'll also add these are all middle-aged shells, so we're not looking at different life stages within them. But things change when we look at the most recent shells. So these are ones that I collected in 2015. And so some of them look perfectly fine and normal, like these three here. That's why I'm showing you more variety, because they're less uniform. The ones in the middle, you start to see really small, like little dots of crystals and different colors. So the different colors mean instead of rotating like this, they're starting to rotate out of plane and come out of the plane towards you. And the only crystal that this actually looks like is when you see wound repair. So if you were to go to a beach, and sometimes you see these like weird scars, they sort of stick up from the shell, actually like a scar would look on our own skin. And that's when the animal puts the shell down really, really quickly to make up for the fact that something has injured its shell and it needs to lay some shell down ASAP before someone comes in, sucks it out of its shell and kills it. And that tends to be an amorphous crystal, so it doesn't have an actual crystal lattice because of how quickly the animal laid it down. And so these regions here and here sort of look like that, like the animal's just putting down shell as much as it can. It's sort of stressed out and trying to do it quickly, as opposed to sort of the slow, methodical growth process that usually results in a really uniform lattice. Okay, and so this sort of goes with a decline in these shelled species in long-term data. So this is a long-term data set. Again, classicists, please don't make fun of me for calling this long-term. But we're seeing that, these acronyms are all different species, but this black bar that dominates at high pH and gets less abundant at low pH, those are barnacles that have a shell. That's the B here. And so we see the same thing. We see that there are more barnacles when the pH, there's more barnacles and algae when the pH is high, and as the shelled species get bigger, so we're seeing fewer mussels here, which are these blue bars. And you see fewer of those as pH goes down. I'm sorry, that graph's a little hard to see at the moment. Okay. So very quickly, I'll give you a second example, and this time we're looking at algae that has a skeleton. It's called crustose coralline algae, because at first people thought it was a coral, but it turns out it's not. It's algae that just has a skeleton to protect it from snails that like to eat it. Thickness is really important to these guys because they like to overgrow each other. If you've ever seen lichens on a rock and they're all sort of flat but overlapping, we see the same thing. And so thickness sort of keeps their competitors at bay. So thickness is a good thing. But we've seen since the 80s to sort of present day that they're getting a lot thinner. This is a real photo of two different specimens taken to scale at conferences. Some smart aleck always raises their hand and is like, did you zoom in the same amount when you took both of these pictures? And I did. It's just very dramatic and very sad. Okay. And so we're seeing this, interestingly, in species that used to be really thick. These solid dots are the historical specimens. Again, they've thinned by half, just like the mussels, which is sort of eerily coincidental. The ones that were thin already haven't changed, which is also really interesting. But those ones that haven't changed on the outside have changed on the inside. So these cell walls. This gray stuff is calcium carbonate shell that's around each cell, and the cell walls of the shell have gotten thinner over time, just by a little bit, okay? But these are all things that are telling us that these animals are having a harder time making their shells, and they're trying to come up with ways that they can get around making less. So the ones that were already thin probably can't get away ecologically with getting any thinner, but what they can do is start to reduce sort of the density of what's going on in their bodies. So I did these transplants, put them really close to each other so that they had to grow on top of one another, and then counted how many times they won against each other, okay? And I made these interaction networks. We're sort of like with the flowers and the bees. The arrows are pointing to who's winning that interaction, and away from whoever lost, and these different colors are species. And so we can see that as we go forward in time, these are computer simulations that I made based on these communities. We can see that the winning dominant player changes from this red guy to the yellow guy. So from a thicker species that I showed you to one that's thinner and not had a change in that. All right. Because they're really long-lived, though, that's not translating to how abundant they are in nature. So this is a long-term data set that, that's ongoing. So with that, I just want to sort of summarize. I know I've given you a lot of different examples of this, but the point is that these physiological observations have led to hypotheses about how ecological communities, how their interactions between species could be disrupted. And I just briefly touched on this, but targeting these interactions and the interaction webs that I just showed you sort of helped to detect potential places, potential weak points in the ecosystem that could be changing. And I'm currently using models to sort of play that forward and fast forward to see what we might expect will happen. And again, that long-term and historical ecological data, sort of historical records, older studies that maybe people aren't reading anymore that can serve as reference points, those can be really important tools for climate science. Cool, thank you very much.